Hi everyone, my name is Kristlyn Lau, and for my journal club, I'll be sharing a 2023 article from the Psychiatry Journal of the American Medical Association about how blood metabolomic signatures and the gut microbiome interact with individuals with major depressive disorder, or MDD. And I've included the citation there as well as the end of the presentation. So why do we care about that? So major depressive disorder is a pretty serious condition that affects various systems in the body beyond just mood. MDD impacts cognition, motor function, immune responses, and metabolism. About 300 million people globally are affected by MDD, with many of those 300 million being unresponsive to standard treatment like antidepressants. We have some current research that suggests depression involves complex metabolic disruptions, especially with respect to lipid and energy metabolism, which is the focus of this study today. So using metabolomics and the microbiome to research new biological pathways for treatment is necessary for the development of a more targeted and integrated approach that ultimately helps with depression. So the infographic that I have included on the screen shows some statistics about just how prevalent depression is. And as significant as these numbers are, depression is often brushed off as not a serious illness. But I would like to bring your attention to the statistic in the orange bubble. Suicide being the second leading cause of death among 10 to 34-year-olds is quite devastating. So the more we study what factors influence MDD, the more we can decrease that number. All right, so let's dive in. The study's main question posed by the researchers was, what are the metabolic signatures of major depressive disorder, MDD? What direction do they take, and how does the host gut microbiome contribute to these signatures? So first they wanted to identify metabolic differences in those with MDD. Then they wanted to assess causality via Mendelian randomization. And this is a kind of study that uses genetics that are linked to the variable being studied. So in this case, they used a genetic variant that's linked to both metabolite production and MDD to see if those with depression have disrupted metabolite levels. And is not going to be influenced by environmental factors since it's genetic. And lastly, they aim to integrate gut microbiome signals to explain metabolic shifts. They ultimately want to know, does the gut microbiome impact shifts in lipid levels in patients with depression? This figure outlines the framework of the study. It shows the relationship that this article sets to highlight, specifically the connection between the gut microbiome and the human metabolome with major depressive disorder. So the arrows in this image depict the directionality of association. The darker shading indicates stronger associations. As you can see, the arrows between depression and the human gut microbiome, as well as the human metabolome, are quite dark. There have been previous studies connecting the other relationships in this graph, notated by the blue arrows and the little summaries on the side there. They are referenced in this study with the blue arrows, but the green arrows depict associations that were not studied nor used in this study for reference. So I've just outlined the goals of this study, and they did make some contributions here. They have identified 124 metabolites that they have found to be associated with MDD. 49 of those had not previously been thought to have been linked with MDD. And then that Mendelian randomization showed causal links between MDD and VLDLs, IDLs, and fatty acids, which are those different levels of density lipids, which I'll get into that in the background. And then lastly, they did note some bacterial shifts in MDD patients in regards to certain groups of microbes, and most of those shifts were linked to lipid imbalances. So some previous studies have found that abnormal levels of lipids, like those HDLs, the high density lipoproteins, the low density lipoproteins, and the very low density lipoproteins, as well as triglycerides and energy metabolites in people with MDD. These changes may contribute to mitochondrial dysfunction, with lower HDL levels linked to more mitochondrial DNA. I did find a large study that found people with depression tend to have lower HDL and higher, very low density lipoproteins and triglyceride levels, though the reasons kind of remain unclear. So they're tying the gut microbiome into this, where it can influence lipid levels and mitochondrial function. So disruptions in gut microbes have been linked to depression. So this study design was a cohort study that used data from UK Biobank. A cohort study is an observational design, and UK Biobank is a publicly accessible biomedical database. They recruited people from the ages of 37 to 73 from 2006 to 2010, and they have over 500,000 people's information. And from those individuals, there were 58,257 participants chosen. 6,811 had lifetime MDD, and 4,370 had reoccurrent MD, where they would get better and then get depressed again and back and forth throughout their lifetime. The rest were controls that did not report any depression at baseline. I'd like to note those with other reported mental health disorders like schizophrenia were excluded. A random subset of individuals from Biobank was profiled for metabolites in their blood using the Nightingale platform. The Nightingale platform is a nuclear magnetic resonance, or NMR, spectroscopy, and it's able to analyze many blood samples quickly for metabolite levels like lipids. For the Mendelian randomization study, they publicly accessed the available summary statistics from a 2019 genome-wide association. They had 59,851 individuals with MDD and 113,154 that were the control. One of the statistical analysis that the researchers implemented was the logistical regression. This is a type of statistical analysis that accounts for differences that may skew results. How this works to test association of metabolite levels with lifetime and recurrent depression, they take into account factors one at a time. So in the first model, they accounted for gender, age, fasting time, race, and ethnicity. And they ran that and they got results for that model. And in the second, they accounted for all of those factors in the first model, but added BMI to see the difference. And so on for the last two models for antidepressants and lifestyle. They also used a false discovery rate to see which metabolite associations were statistically significant with MDD. So with all of those statistics and the 4 different models that they observed for MDD versus control, they did find that those with depression were significantly younger, with a median age of 56, versus 58. They were more often female, 65% to 35% male. They had a higher education, higher BMI, less physical activity, consume less alcohol, and were more likely to come from a Black or mixed racial or ethnic background. So all of these things have been accounted for in the results. So their metabolome association analysis showed that even taking into consideration these variabilities, the percentages of metabolite levels associated with MDD that remained significant were pretty substantial. So we started at 71% and we landed at 49.8%, showing that the metabolites they tested for were still significant. So among the 249 metabolites that they tested for, they determined that 124 of those were associated with MDD. They span energy and lipid metabolism, with 49 of these metabolites being novel associations. Nobody else has made this connection before. Notably, citrate was decreased and pyruvate was increased, suggesting a bioenergetic disruption with those with MDD. So the results of the Mendelian randomization suggested that certain fatty acids and lipoproteins causally contributed to MDD risk. The ones here were increased levels of VLDLs, IDLs, and other fatty acids. It was a little inconclusive for HDLs and citrate-related metabolites. Those changes appear more like downstream effects. This could possibly be due to the strong influence of diet on the microbiome. So the gut microbiome associations that they have found were mostly bacterial shifts increasing and decreasing specific taxa. So the decrease was mostly found in beneficial bacteria, the bacteria that are linked to the more favorable lipid levels. So we've got Proteobacteria, Bacteroidetes, Ruminococci, as some of the beneficial taxa that were less prevalent in those with MDD. And for the increase in harmful taxa, we've got Flavinofractor. I'm so sorry if I mispronounce these so bad, but you guys get the point. Most of these shifts in the levels of the microbe taxa were linked to those lipid imbalances, especially that increase in the very low density lipoproteins and the monounsaturated fatty acids, with a decrease in those high density lipoproteins. We'll see a little bit more of that in the figures. So this figure shows how specific metabolites correlate both with the gut microbiota profiles and MDD. This suggests like a triangle of influence with microbiome, metabolites, and depression. So the microbiota produce the metabolites and have an impact on depression. These scatter plots show this correlation specifically between the metabolic signatures of microbial taxa and MDD. The taxa in all of these plots are negative correlation. So they show, I know it's tiny, but each dot represents a metabolite. So we've got the HDLs, the VLDLs, and so on, fatty acids, small molecules. They're in a pretty straight line there, but I'll break down. The x-axis shows the association of the metabolite with microbiota taxa, and the y-axis shows the association of the metabolite with MDD. So the different colors, again, represent different classes of metabolites. I'm not sure if you're able to see, but there are some that are colored or that are circled in black, and those are the ones that are specifically associated with MDD. You'll notice the numbers say Z-score on the bottom and on both axes. These include the results of association analysis of gut microbiome with 16 sRNA sequencing and with the Nightingale metabolites. But this just shows that these specific microbiota are inversely associated with MDD. So this figure is very similar to the previous scatter plot, but this contains the classes of microbiota that have a positive correlation between the metabolic signatures of microbial taxa and MDD. The x-axis, again, is the association of the metabolites with microbial taxa, and the y-axis shows the association of metabolite with MDD. Again, the different colors represent the different class of metabolite. So you see what the metabolites produce have an impact on MDD. So overall, this study had strong evidence that major depression is linked to metabolic and microbiome alterations, specifically depressions in lipid profiles, mitochondrial dysfunction, gut dysbiosis. And citrate is the interesting one here, possibly serving as a key metabolic hub connecting all these pathways. I would like to notate some strengths of this study. This is the largest to date study on metabolite depression links, and the controls for confounding lifestyle, medication factors is a huge plus. So the data is less likely to be biased or skewed. They also use that NMR spectroscopy for precise metabolite detection, and they integrated those metabolomics and microbiome analysis pretty convincingly. Some limitations of this study were the cohort observational design. It doesn't actually confirm causality. It's just a lot of statistical analysis that confirms a probability. The blood metabolites don't always equal brain metabolites, and the microbiome data that they got to compare was from a separate cohort. It wasn't the same amount of individuals. It wasn't the same exact individuals in each, the Mendelian randomization and the microbiome analysis. So there are some clinical and research implications here. We learned that there is potential for metabolomic biomarkers to be used with MDD, possibly be able to detect MDD or even microbiome targeted therapies. But we still don't know. We don't have enough information because it was an observational study. It had amazing statistical analysis, but we need interventional studies with a more robust design. So I have some discussion questions here. How might metabolomic profiling improve the diagnosis or subtyping of major depressive disorder? So could these metabolic markers help identify biological subtypes of depression or even predict treatment response? So this study found that certain metabolites likely cause MDD while others are consequences. How should this distinction guide treatment strategies? Should interventions target causal pathways only, or should they treat downstream effects also? Could that be a therapeutic option? Third, given that the microbiome data was from separate cohorts, how strong do you think the evidence is that the microbiota directly influence depression-related metabolites? What would an ideal experiment design look like to confirm these links? And then, can lifestyle or dietary interventions realistically modulate the specific metabolite and microbial signatures identified in the study? Are probiotics, prebiotics, or natural strategies a viable option based on the current evidence? What do you think? And lastly, what ethical or practical challenges might appear using metabolomics and microbiome data in psychiatry treatment? For example, concerns about data privacy, cost, or ethical implementation. These are some of the references that I used in my presentation today. Thank you all so much for your time and happy journaling. I look forward to see what you all have to say. Thank you.